Okay, so hello once again, everybody. Welcome to the people who just joined. This is your host for today's webinar, Zoltan Perestegi from RotaChrom. If you're new to our webinars, you're welcome to listen to our previous presentations online. All you need to do is visit webinar.rotachrom.com and register there to watch all the previous webinars. As you most probably know, RotaChrom manufactures the world's only integrated industrial scale centrifugal partition chromatography, or CPC, instruments. Established in 2014 in Europe, RotaChrom has presence all around the world with applications in the pharmaceutical, natural extracts, food and beverages, and cosmetics and fragrances industries as well. The speaker for today will be Arpad Konczol, PhD, head of RotaChrom's research lab. And yes, RotaChrom actually, although we're an equipment manufacturer, we do have an in-house research lab. So let's hear a little bit more about that from Arpad himself. And also, please use this opportunity to ask him as many questions as you have or as possible. There's a Q&A button at the bottom of your screen. You can type your question there, and we'll try to answer as many questions as possible at the end of today's session. If we don't get to answer your question, we'll follow up with you directly. With that, Arpad, welcome to our webinar. The virtual floor is yours.
Thank you, Zoltan, for the nice introduction. Ladies and gentlemen, thank you for joining us today in this webinar. My name is Arpad Konczol, and as Zoltan mentioned, I work as the head of the research laboratory at RotaChrom Technologies. Basically, I'm an analytical chemist, and I have a PhD in pharmaceutical sciences. At RotaChrom, I'm responsible for the R&D activity and collaborations. So basically, I am the ultimate tech guy at the company. Today, I'm going to present you the pesticide removing capability of our CPC technology. First, I would like to highlight the importance of the issue of cannabis contaminants with special focus on the risk and regulations associated with pesticide residues. Afterwards, I will shortly touch on two of the principles of the CPC technology. And based on these 2 points, I will present you a case study in the remediation of cannabidiol, the pesticide remediation of cannabidiol, or CBD. And finally, I will give you and provide you a general strategy how to manage the purification of phytocannabinoids from pesticides. I will also call your attention to our next webinar provided by my research fellow, Dargo Gergő, and he will provide you details on the green and sustainable features of centrifugal partition chromatography. I guess it's more than trivial for the audience that pest management is an ancient topic and practically is a must in modern industrial crop cultivation to avoid plant diseases and to maintain high yield mass production. On the contrary, a lot of regulatory, safety, and environmental issues should be considered when discussing this complex topic. Please note also that the term pesticide is an umbrella term and covers a myriad of substances that can be classified by use, origin, or chemical structure. Turning to one of the most popular crop nowadays, to the hemp industry. So the pest management in the hemp industry is really controversial. It's also a difficult situation, not just for the cultivators, the regulators, but also for the consumers. Some points to emphasize this situation. Since cannabis products are not only administered in edible forms, but also smoked and inhaled, unlike most agricultural products, residue pesticides pose an unpredictable risk to consumers. Let me just mention the potential role of microbutanil in the recent vape crisis. Unfortunately, federal and state laws are still conflicting on cannabis-related pesticides. However, the good news is that 10 pesticide products have been registered specifically for hemp by the US Environmental Protection Agency. In addition, I have to also mention that the cannabis plant can very efficiently absorb contaminants from the soil. It's also called the bioaccumulation and also used for the purpose of phytoremediation. When we're considering a typical process flow of, in the cannabis industry, I don't want to go into the boring details, but my advice is just to monitor the levels of pesticides in each of this entire process chain with an appropriate analytics. And the question arises, what has to be done with the contaminated biomass and products? At RotaChrom, we set ourselves the goal to develop a strategy based on CPC to remedy this situation, this complex and difficult issue. Currently, there are 3 compendiums in force in regulatory areas of Oregon, California, and Canada, in which pesticides to be monitored and action limits thereof are declared. As the most extensive and strict regulation is provided by the Canadian list, we have used this one as a reference in our studies. And just to illustrate the real world situation, I just picked this test results from one of our clients in California. On the left-hand side of the slide, you can see the test results showing that 11 pesticides were detected and 6 thereof exceeded the actual action limits. The most frightening, however, regarding in this sample, that this contained a huge amount of carbofuran, a category 1 substance. It's better not to think the potential toxicological hazard of this material. Okay, I guess in the previous series of this webinar, we presented that CPC technology is a straightforward, high throughput and industrial scale solution for the efficient separation of phytocannabinoids. On the left-hand side of this slide, you can see an industrial scale separation of CBG, CBD, CBN, THC, and CBC. In any case, the question arises, this is really nice, but where are the pesticides? So to answer this, we set ourselves the goal, as I already mentioned, to fully characterize our pesticides removing capability of our methodologies. Our results in this topic was already presented 2 years ago before the pandemic in a prestigious chromatography conference in Baltimore, and I'm really proud to represent that this work was acknowledged by a prestigious award just to proving the relevance of this topic. On the next 2 slides, I would like to recap to the theoretical background and the definition of CPC. So CPC basically is a unique preparative liquid chromatographic technique in which the separation occurs between 2 immiscible liquid phases. The stationary phase in CPC is immobilized inside the rotor by a strong centrifugal force. And the mobile phase containing the sample is fed under pressure into this rotor and pumped through the stationary phase in the form of tiny droplets. So basically in CPC, the chromatographic column is the rotor. And the separation and the retention of compounds is simply based on the partition of each and every compound. So in this definition, I would like to call your attention that compared to conventional solid support utilizing chromatographic techniques in CPC, the role of the phases, I mean the stationary and the mobile phases, can be easily switched. And this is a huge freedom compared to conventional chromatographic techniques. Another theoretical point is, which can be utilized in the method development, is the simplicity and the relation of the partition coefficient and the resulting CPC chromatogram. I mean that the partition coefficient of the compounds in the mixtures to be separated is directly correlated with the elution profile of in the CPC chromatogram, and this is also true vice versa. And this is also a huge advantage in the efficient method development in CPC. Okay, after this short chemical part or technical part, I just put all of the chemical structures of the Canadian list, and with the two major cannabinoids, CBD and THC, on this slide, just to illustrate the diversity and the polarity range covered by these substances. So basically, in this Canadian list, 150 compounds are declared. And as you can see, a really wide polarity range is covered by those compounds. So we can easily see that this is a really challenging separation task. And coming to the case study of CBD, in this slide on the right-hand side, you can see a heat map, a so-called heat map, just representing the CPC fractions in the vertical axis and the tested pesticides in the horizontal axis in an ascending order of partition coefficient. So in this study, we just spiked a CBD oil with all of these pesticides listed on the Canadian regulation, and used our baseline methodology to get this elution profile. CBD is marked with blue in the middle of the heat map of the chromatogram. It's easy to see that the vast majority of pesticides eluting before and after of CBD, and there are some compounds that are co-eluting with CBD, and there are some examples of so-called outliers which showed an irregular partition coefficient and elution time correlations. But I'm really proud to present you that 19 out of these 150 compounds are fully removable in a single and generic CPC run. So afterwards, just to mimicking a real industrial scenario, we performed the so-called overloading studies just to acquire the isotherm of CBD in this solvent system. In the chromatogram, you can see that just increasing the concentration of the injected CBD oil, the peak of CBD corresponding to CBD became fronting, and the apparent retention time shifted to the right to the higher retention times. It means that the apolar pesticides are more prone to coelution with cannabidiol. Okay, and just to eliminate those problematic coeluting pesticides, we, and following also the industrial scenario, we used the so-called sweet spot approach, which is basically a rule of thumb published by the father of CPC, Yoshiro Ito, Professor Yoshiro Ito. And this approach states that the highest resolution in the given CPC system can be exploited if the KD value of the target compounds fall in the range of 0.4 to 2.5. So according to this approach, we have to adjust the solvent system composition to get KD values in this sweet spot range, and that's what we performed in this case study of CBD isolation. And here are the results for the problematic pesticides. On this heat map, you can see also the elution profile in the CPC run in ascending order of KD values of the target compound CBD and along with the pesticides compounds. And here you can see that this simple modification in the solvent system significantly improved the resolution of the method, but some coelution still remained. And what about with the apolar pesticides? In this special case, the solvents, the water content of the solvent system has been decreased, and the mode of elution has been changed from descending to ascending mode. And as you can see, the majority of these problematic pesticides was also resolved from CBD. Okay, so coming to the final results of this case study with CBD, we also studied the most frequently used pesticides in this content. And here you can see the results in the three regulatory areas: Oregon, California, and Canada. On this pie chart, you can see that the most frequently used pesticide residues can be fully removed by this simple and generic CBD methodology that we have developed at RotaChrom. And I wanted to call your attention that, for example, microbutanil and bifenazate, most frequently used in all of these regulatory areas, were also fully removed. So coming to the final strategy, the workflow starts with a reliable and precise pesticide contamination profile that has to be acquired of the cannabis sample. This is a prerequisite. Then, if it doesn't indicate problematic impurity material, the material can be purified by the baseline method. However, if quality pesticides are present in the input sample, basically three ways can be followed: either by adjusting the fraction collection, the critical pesticides can be eliminated, but I have to emphasize that in this case, the yield will be compromised. As I already presented today, in the second case, if we are using the so-called sweet spot approach, satisfactory resolution can be achieved also for these problematic ones. And finally, I have to add that a third approach is also feasible. This is the so-called computational guided solvent system selection approach that is developed by one of our academic collaboration partners in Germany. So as a summary, I wanted to share with you that at RotaChrom in the research lab, we have developed and validated the CPC-based remediation of phytocannabinoids, and we worked according to the most stringent Canadian list. The main conclusion of our work are that pesticides can be easily separated from CBD by our technology, but there are some polluting and problematic ones, but we have also developed an approach to get rid of those ones. And I wanted to add and emphasize that the presented methodology can be easily extended to other input materials and target compounds such as THC, CBG, or other cannabinoids. So the final message from today's presentation, today's webinar, is that this comprehensive and experimentally validated know-how for pesticide remediation is ready at RotaChrom. And I do really hope that I managed to demonstrate you that even such a challenging chromatographic task can be tackled by means of CPC. Thank you for your kind attention, and I'm more than happy to answer all of your questions related to this specific topic, or in general questions related to our technology.
Thank you very much, Arpad. Yeah, I hope Arpad's presentation actually demonstrated that RotaChrom is not only manufacturing the device or the machine itself, but we do have the know-how as well. Work with you on your specific chemical separation problems, including remediation of pesticides. So as I look through the questions, until I do that, actually, I would like to draw your attention to our upcoming webinar. So feel free to register now. You can do it already now at webinar.rotachrom.com. It will be about the green features of centrifugal partition chromatography. And sustainability is a key business requirement and challenge these days. So I hope that more of you or many of you will register. And yeah, don't forget that you can forward the invite to your coworkers as well. So now let's see the questions. I see a first question here asking about the federally approved 10 pesticide products. Do you have any experience with those ones in your methodology is the question, Arpad.
Yeah, thank you, Zoltan. So as I already mentioned in the introduction, 10 pesticides products have been approved specifically for the use in cannabis cultivation. And the good news is that those are typically 9 of them. Those 10 ones are typically biopesticides composed of neem oil and garlic oil, and some contains also fatty acids. But 4 of those 10 one contains the chemical substance azadirachtin, and which is easily removable in the CBD isolation methodology. So we have expertise with these substances also.
Okay, I have a follow. It's a different question here, asking about the levels at which this process has been checked, asking about CBD recovery.
Yeah, this is a really good, relevant question. I mean, as I presented, if the contaminant, so the pesticide residues are in polarity far away from CBD, if they are more polar or more apolar than the target compound, then the resulting yield of the pesticide remediation technique will be not compromised. But in the case when we have problematic ones and co-eluting ones, then a small compromise on the resulting yield of CBD has to be taken into consideration. And let me just add that the levels of the levels of validation of this approach, that we have validated this approach at 3 stages. First, in the just measuring the partition coefficient of all of those pesticides on the Canadian list and the corresponding cannabinoids. And afterwards, we have performed a lot of spiking experiments and CPC runs to get this know-how and validate this approach.
So the next question is actually about the scale. So yeah, I guess you can mention about the scalability of the different platforms of RotaChrom, but yeah, the question is, what scale have you been able to achieve?
Yeah, the answer is that the huge technological advantage of the RotaChrom CPC platform is the scalability. So we have checked this approach on our smaller machine on the RCPC with a total rotor volume of 2 liters. But thanks to this cylindrical shaped geometry of our cells and the scalability, all of the results that we obtained in these studies is fully transferable and predictable for the large scale, which is capable to purify multi-kilograms per day of CBD.
Thank you. There's also a question about, so I'll just read the question, sorry. Did you manage to purify the heavily contaminated THC distillate showed in the introduction?
Yeah, unfortunately, I just omitted this slide because it's one case study is more than enough, but I can show the chromatogram that we that we obtained in this study. Can you see right now my screen?
Yeah, we see it. Yep.
Okay, great. So in this chromatogram is acquired with that sample, with that heavily, extremely contaminated THC distillate. And here you can see that most of the pesticides are eluting before the peak of THC, and there are only one pesticide, namely piperonal butoxide, which is problematic and co-eluting with the target compound THC. But I have to emphasize that in this run, just adjusting the fraction collection, the limit, so the level of this pesticide was also decreased below the corresponding actual limit.
Thank you very much for answering that one. Somebody's asking about the analytics needed for proper pesticide testing. So what kind of analytics is needed?
Yeah, this is also a really novel and popular topic in the testing of cannabis samples. I would say that most of the analytical vendors who are providing not just instruments but analytical methodologies for pesticides testing can be a solution. I would say so. Basically, HPLC and mass spec is needed for the sensitive analysis of these pesticide residues, and in some cases, GC, a gas chromatograph coupled to a mass spec, have to be applied to acquire the concentrations and the level of contaminations of those pesticides.
Thank you. There's also a question about the input purity of the CBD where the process was applied. So they're asking about pure. Well, was it pure CBD, raw CBD? I suppose that's a question about the input that for the case study.
So in the case study, we used the CBD distillate. Approximately a CBD content of 60%, but the same approach can be easily applied for a crude extract so the robustness of the CPC technology can be managed all of the matrices in the downstream process of cannabis samples. So crude distillates, whatever.
Yep, thank you. Thank you for answering that one. We have a question about the situation in Europe because your data has been, or the standards mentioned, you focused on North America. So do you have any information about the situation in Europe?
Yeah, so in Europe, this pest management is a different scenario, I would say. In our studies, we focused on the US states and then on the Canadian list more specifically. But I would say that the approach can be extended also for other target compounds and other pesticide residues. So the approach that we developed is is a universal one. And let me just add that in Europe, not just the pesticide residues, but also the heavy metal contamination of cannabis and hemp, more specifically hemp samples, is an issue.
Thank you. One of our listeners is asking if you achieved pharmaceutical grade purity, which they define as larger than 99%.
I mean, in general, if the target is of the CPC run is to get a really high purity, it's achievable. But again, this is always a compromise between throughput, yield, and purity. So I would say that after in the CBD isolation downstream process, also the crystallization of CBD should be considered as a more feasible downstream approach than directly using chromatography for the isolation.
Perfect, thank you. And the final question that we have for now is about the scale, kilograms per run or kilograms per day. I guess you can give for approximate numbers for both of the instruments.
Yeah, for sure. So if the question is related to the CBD isolation capability of our systems, then I would say on the smaller machine, 100 grams, approximately 100 grams, but it's heavily dependent on the input material, so the composition of the input material. But I would say that 100 grams per run, which is typically 20 to 30 minutes, can be managed in CBD isolation. And this is also valid for our bigger system, for our ICPC unit with a total rotor volume of 23 liters. In that specific case, approximately 300 or 400 grams can be injected in one run, and some kilograms can be processed in a day.
Okay, I don't have any more questions. So with that, thank you once again for your time and attention. We really hope that you found today's session useful. And if you're watching the live session now here, we will send you a link to view the replay of today's session shortly. And don't forget, if you would like to dig deeper into today's topic, you're welcome to join our monthly podcast called Purified, where I will interview Arpad just now, which will be available in about a week's time, and discuss today's topic further. You just need to search for Purified by RotaChrom on your choice of podcasts, or find the link to the podcast on RotaChrom.com. Also, don't hesitate to reach out to us at sales@rotachrom.com should you have any questions, or you'd be interested to learn more about our purification platform. And please continue to wash your hands frequently, wear a mask, even if you're already vaccinated. And I hope to see you again on May 20th. So don't forget to register now at webinar.rotachrom.com. Enjoy the rest of your day. Thank you.
Thank you.